o'clock in Europe and 11 o'clock on the East Coast of the US. We are getting started on time. Today's webinar is called Preclinical Assessment of Targeted Radiotherapy, a step further toward precision medicine. To begin, I will address a few practical questions. The first one, we are recording the webinar. The presentation will be available to all attendees afterwards. Second, we have prepared a series of polls. The first questions will appear right now on the right-hand side, on the left-hand side of your screen. The first question is, in order to adjust the content of our webinar, rate your knowledge level about TRT. I will leave the survey for 1 or 2 minutes and will be to read your answers. Third, after the presentation, Cyril will answer a few questions. So if you have any, please type them in the chat panel. We will gather all the questions at the end of the presentation and Cyril will answer them to you. Finally, if you're experiencing technical difficulties, please ask directly the chatbot on the right-hand side of your screen. Now, let me introduce you to Cyril Berthet. Cyril holds a PhD in molecular and cellular biology from the University of Lyon in France. In 2002, he joins the mouse cancer genetic program at the National Cancer Institute in Fredericks, US, as a research associate. Cyril joined Oncodesign in 2007 as a project leader. And today he is our pharmacoimaging unit director. He is also the director of PharmImage operations. Cyril, are you ready to start your presentation?
Yes, thank you, Krista, for the introduction. So today we're going to talk about the preclinical assessment of targeted radiotherapy, and I will give you first some background on what is radiotherapy. So just a quick overview of what will be the topics today. A short introduction of the targeted radiotherapy nuclei itself, TRT, to be more shortcuts, why TRT as a theranostic agent, and the current progress in TRT, notably in clinic. Then I will quickly overview our capacities at Oncodesign, what we can do as a service provider, starting from the chemistry, the in vitro evaluation, and the in vivo evaluation of TRT. And then I will move back to a more background aspect. Can TRT be combined like radiotherapy? Is it the same thing? Should we address differently such kind of combination? How it is done in clinic? Different question that may you go beyond just the simple therapy. And finally, a short slide on PharmImage and our network. So as mentioned by Krista, you have some poll, and so the first one is just to highlight what kind of expertise you have, and I can see that most of you are beginner, which is probably good for this seminar. I hope I can give you some good background. So starting with what is targeted radiotherapy. Targeted is the combination of radionuclides that is attached to molecules that has a specific affinity to tumor cells. Usually we are talking here in oncology, TRT is meant to target oncologic cells in most of cases. It could be any kind of molecules. It could be an antibody, it could be a fragment of antibody, a proteins, a peptide, or even small molecules. And you will attach through a chelating agents or covalently link a radionuclide. You have two types of particles. This nuclei will deliver a specific energy, and it could be derived from alpha particles or beta particles. It can make a big difference what it is within the body. The alpha particles are more energetics that will drive more DNA damage, but they're also much bigger, and the length of penetration in the tissue is usually in the range of 50 to 100 micromolar. As an example of alpha particle, you will see the bismuth, the radium, the actinium. The last one, actinium, is being the mostly used right now. And then you have the beta particles, which are much smaller, and you have different kind. The most famous one that is well used, and the only one that is in clinic, is the lutetium 177. The length of the lutetium is right of the penetration in tissue quite short, is below 200 micromolar. Then you have other isotope like copper 67. You have so the indium 131 that is currently used in thyroid and in many clinic, and you have also some isotope that have a much longer length like yttrium 90, which sometimes could be very good if you have a big tumor, but also for toxicity. You might have more side effects. So you have to decide what is the best according to your indication, according to the molecules, what is the best nuclei to be used. And this is not an easy question. If the good thing about TRT and beyond the isotope is that some of them, the one I just highlight, have a therapeutic effect. But most of the time, we can exchange the isotope that is used for therapy with another one that is used for diagnosis. And the emission here will be a gamma emitter that you can see with the PET camera or SPEC camera. This is very, very important as a diagnostic agent, because then you can do the diagnosis and you can do the therapy together with the same molecules, which allows you to do a treatment stratification of patients. You can first do the images, like it is here on the panel A. You have a patient you have a PET scan with the gallium-68 of the tatate, and you can see all the tumor that is visible. Then this patient will be treated with lutetium dotatate, and we can do here as well an image, but with lutetium the image is a quality that is much lower than gallium, and to SPECT images rather than PET. And on the last panel, you have the patient after the treatment with the lutetium 177. So beside the fact that this treatment is very efficient, we can treat only the patient that indeed has an uptake of your molecules. Clearly, we are in the way of precision medicine. It increased the efficacy for the refractory and inoperable metastatic cancer that can really target through your molecules, your antibodies, or your small molecules, and you really address the isotope only on the tumor that is visible initially by the PET scan. Here there is for the one who know the field, this image is a very classic one. Should I use alpha or beta emitter? Here you have a patient initially that is treated by PSMA, and the first image is with gallium-68, and this patient has a strong signal within the prostate and metastasis, bone metastasis. And when you treat with PSMA and lutetium 177, in this case you have a resistance. There is no there is no effect of lutetium PSMA for this patient, unfortunately. But when they move to actinium 225, which is then in that case alpha emitter rather than a beta emitter, here the patient was sensitive. And we don't know why. Basically, the target is the same, the molecule is the same, it's only the isotope. Why in such case there was a resistance to beta emission rather than alpha emission? This is unknown, and we can really not clearly define yet a proper answer to this question. What is the current progress in TRT clinic now? there are 3 agents that have been approved by the FDA in the last 5 years, which is more than in the last 20s. The Xofigo, which is barely a targeted therapy because it's only a salt that is bound with radium-223, and that is used in the castration-resistant prostate cancer with bone metastasis. The Lutathera, which is clearly the real first one, TRT, that targets the gastroenteropancreatic net cancer that are positive for somatostatin receptors. Then you have the Azedra, which targets the pheochromocytima that express the whole phenylephrine transporters, and that is labeled with iodine 131. There is a major difference between the iodine and any other isotope. The first one is covalently linked to the molecule rather than the lutetium or yttrium or any other that I mentioned to you are metals, and you need a chelating agents to bind to your molecules. I will address this question right after. It is estimated that the market of nuclear medicine with its compounds and the new ones, the PSMA that will come most likely, will clearly increase the market of the radiopharmaceutical in the next few years. And as a sign, there is now more than 50 clinical trials that involve TRT only with lutetium, and most of them are focusing on dotatate or PSMA, which is now in phase 3, and we expect the results at the end of the year. Dotatate has been approved in 2017, and here you have a small graphic when you can see that the response of the patient compared to the control arms is very impressive. Same thing with the PSMA. Here the readout is the PSA level, the blood level of PSA, and you have very strong response, decrease of the PSA blood level for the patient that have been treated with PSMA. And clearly, we all hope that this treatment will be successful in phase 3 and reach the market, because prostate cancer is a major impact on health of people. Oops, sorry, moved too fast. So now I will just overview what we can do, what needs to be done, and what we can do to evaluate the targeted radiotherapy. You need all along the process, you need different expertise. The first one, you need to talk to the chemist to be able to do the bioconjugation and the radio labeling itself. Then there is an in vitro steps which allow you to determine if your compounds, what is the affinity of your compounds to the to your targets, could be done first on the protein itself, but then on the cells, on your tumor models, on slides of tumor model, but also on biopsy of patients. Then you will do the biodistribution of your compounds. It is very important to evaluate if how much your compounds will go to the tumor, what is the off target, if you have some uptake in other organs where you can have toxicity afterwards. And of course, evaluate the efficacy of your compounds before you go to first in man. In parallel of the therapeutic compounds, it is very important to develop at the same time the imaging companion diagnostics, because none of these molecules will be given to patients without knowing if the target is indeed expressed. You can imagine that if you administrate radioactivity to patients, it's better than to know that indeed you will have a positive uptake of your compounds. So starting with chemistry, here I took an example with an antibody, but it could be a peptide or even a small molecules. First thing is to do the conjugation, and you will use a chelating agent. You have different kind. I will show you the next slide, and then once you have validated that your conjugation is stable and do not, in fact, impairs the property and notably the affinity and selectivity of your compounds, you can move to the radiolabeling. This is important to evaluate to take the time to evaluate the proper bioconjugation, because otherwise none of your in vivo experiments will be reliable. And as I say, if you select well your chelating agent, you can switch from a therapeutic isotope to a diagnostic isotope. Here is an example of lutetium that you can switch with gallium-68. You will evaluate the purity, the stability, and the specific activity, which is an important criteria in nuclear medicine. Always moving to slide, sorry. This is the different parameter that we have to take into consideration. What is the PK of your compounds versus the half-life of the isotope? Using isotope half-life of 2 hours, if your protein half-life in the blood is 3 days, makes no sense. So we have to adjust to be able to detect the compounds as long as it is in the body with the proper life of the metals, or the other way around. If the half-life is very short, it makes no sense to use a metal or a radioisotope that has a long life where you will irradiate the patient for no reason. So once you have decided which isotope you want to use, you need to work on the specific activity, how many chelating agents you should add, what will be the loading of your chelating agent with the isotope to be able to determine your specific activity. If you have a poor specific activity of poor loading, there is no chance to get an image or even a therapeutic effect. So it's important to determine that. You can play on the condition, on the heating, on the pH, on salt buffer to optimize the number of isotopes that will bind to the chelating agents. So what we call the DOL is the number of chelator per proteins that you will add. Usually a good number is around two chelating agents, which will allow you to have a good specific agent. Of course, it depends if you're doing a site-specific technology or if you're doing a random labeling. Random labeling for any research is usually fine. If you want to go to clinic, you will favor site-specific, just because it might be easier along the way to prepare your GMP batch if you have a site-specific bioconjugated compound rather than random, where you will have to characterize at every batch. So there is a second question just pop up. See if you have molecules that if you want to investigate, you can answer the question, and we'll be happy to give you some help on the topics. Here is an example of a study that has been performed here, where we play with different different chelating agent, DOTA, DOTAGA, NODAGA, MANOTA. I won't get into the details of the chemistry, but you can see on the first panel that with the MANOTA we were able to incorporate a much more isotope than we did with the other chelating agents. On the other hand, with the MANOTA, the KD was lower than with the other. So it's a compromise. Do you want to have a better specific activity with a lower affinity, or the other way around? So when we move to the in vivo biodistribution, we observe a much better tumor liver ratio with the MANOTA, which is very important in that case. And if you look at the image, here you have a DOTA with an uptake in the tumor, but an uptake in the tumor which is more or less the same in the liver, and an uptake in the liver will drive some toxicity. Same thing for the kidney, and this is something that you might want to avoid. Whereas if you use the MANOTA, you avoid the uptake in the liver, and you have the concentration within the tumor will be much higher. So if you consider that in the kidney the elimination will be fast, then chance of toxicity will decrease. This is another example. Here it's related to the bioconjugation step itself. We classically use anti-reactive big functional chelators to attach the chelating agent to the molecules, and we will label that you label with different isotopes. Here, the example is done with zirconium 89, and this is the kind of picture that you classically have, where you will have a metabolism of the chelating edge of the binder of the linker of the chelating agent, and the zirconium will be free, and you will find it in the bones beside the tumor. With using a POTS you attach with the malamide, then you can have a much cleaner image with just zirconium that is uptaken in the tumor, just because you avoid the cleavage of the linker. So again, you have to play with the chelating agent, with the linker, with the bioconjugation strategy to decide what, in your context, what is the best strategy to do the in vivo and clinical study. So there is a whole toolbox to play with, depending on your isotope, depending on the chelating agent that you want to use, depending on the conjugation that you have access to, and the biologics you work with. We have to combine all this together to find the proper molecules to be able to use as a TRT. Here is the lab that we will perform where we perform the bioconjugation. We have different labs. One is driven to fluor 18, and we can do also GMP for fluor 18, and we have 2 cells also for the metallic radionuclide lab. Fluor 18 is dedicated to imaging, whereas the other can be used as a therapy. We are working very closely with kinetic, which is a well-known company that develops and sells all the chelating agents. So we have a strong background to help you on the chelating agents. Now, just to highlight the in vitro evaluation of the targeted therapy, this is a very powerful tool to select your models or even the population of patients that express the target of expression. Here, what we do usually, we take some slice, or we can do cell lysate as well, where we will do the binding of the radioactive molecule alone, and we do a competition with the non-radioactive molecules, which allow us to define the specific and the non-specific binding, where we derive a curve fitting determine the binding affinity, the KD, and the binding of the maximum binding of your compounds. Usually for good compounds for TRT, it's good to be in the nanomolar range. It allows us also to evaluate the selectivity, because you can challenge with other molecules to be sure that you hit the epitope or the target that you want to hit by challenging with other competitors. And having access to clinical samples or preclinical samples, you can do that on your models. You can do a bunch of models or slides of models that you can to select which one you have the best binding, and then you expect the best uptake in vivo. Or you can do also as well for clinical biopsy and select out of 10 for an indication. You can take 10 different biopsies, and you can look if for this indication you have a strong binding or not, and if you have to refine your population before your clinical trial, or if you can move on. There is another question I just actually missed. It was what will be your preferred, and I can see that so far most people will prefer the lutetium 177, which is not a surprise, which is because so far it's the one that is the more available. Then you will do the in vivo biodistribution. It helps you to define first of all, the uptake in the tumor, but also, which is very important, the uptake in other organ and the toxicity that you might expect. Having a target that is very, very selective to the tumor is sometimes rare, and it's very important to be able to assess early on which tissue you may have some toxicity. And when we do such study, we do a kinetic over time. Here it's 72 hours because here we have used indium. But depending on your isotope, we will refine the kinetic. And of course, we always correlate that with ex vivo gamma counting. We collect the tumor on the different organs to be sure that what we observe by PET or SPECT, we can correlate it by gamma counting, which is more sensitive. Then the efficacy itself. Here I just put two examples. It's a compound that is labeled with lutetium, and after one treatment, we observe a complete response. And this is something that we do observe with many other many compounds. This is some if the selectivity and affinity of your compound is good, you have good chance to reach a complete response of your tumor. And when for the same compound, when we did two treatments, dividing the dose per two and 7 days apart, we observed even a better response. So playing with the schedule, playing with the dose, playing with the specific activities, what we call the ratio of the cold compound versus the hot compound, which is very different than the standard molecules, will help you to define what is the best regimen that you should give to human after your preclinical study. Can TRT be combined like radiotherapy? We know that radiotherapy is used in more than 50% of the cancer therapeutic regimen, and it increases the DNA damage in the targeted tumor cells, and it modulates also the tumor microenvironment. And basically, this is what we see observe as well with TRT. That we do a localized therapy or a TRT, we will observe the same kind of effect. The major difference is here you can expect to target the whole body, all the metastases, which is more difficult to do with the standard radiotherapy. The effect that we observe are usually related to cell death, to immune cells infiltrations, necropsy, and apoptosis, and modification of the angiogenesis. But there is a whole bunch of effect that is observed, and doing combination with the therapy may enhance your overall activity. Here is an example of, in preclinical, the benefit of radiotherapy with immunotherapy, the CT26 models. We have treated, in this paper, they have treated with 5 times to gray and associated with PD-1 or PD-L1. And you can observe on each of the treatment, PD-1 or PD-L1, you have a strong synergy of the radiotherapy with the immunotherapy. And now there are several clinical trials that try to combine immunotherapy with radiotherapy. Same thing here. If you use the Zevran, the anti-CD147 with yttrium-90, and you combine with the chemotherapy, we observe in blue synergy, and if you do twice the radiotherapy, you have even a greater synergy. So combination of targeted radiotherapy, there is a good chance that we will observe what has been shown previously for the last 20 years with radiotherapy, and this is something that you clearly have to keep in mind in your development. This is a standalone therapy, but combination will definitively come right after. And to illustrate this, the dotatate, which is now given as a standalone therapy for the BEDNet tumor, there is a clinical trial now that combine the dotatate with 5-FU, the dotatate with capecitabine and temozolomide, and even two dotatate, one with lutetium and one with yttrium. And this one is illustrated on the bottom panel when with a combination with 1 of 2 cycles with the yttrium, then followed by 2 to 3 cycles of lutetium. You observe a reduction by 67% within the 3 months, and 21 out of 26 of the patients were stabilized, and 7 had a partial or complete response. So the combination dotatate has been approved in 2017, and now clinical trials in phase 2 are starting for the combination. There is another one here, the Zevran, which has been approved much earlier. So it's yttrium antibodies as well. And here it has been combined with a high dose beam to chemotherapy and SCT, and you can see from the paper published by Shimoni that clearly you have an improved in this clinical trial of the combination beam alone versus beam plus Zevran. We don't know here the Zevran alone in this clinical trial. But clearly this is something that probably is a field that we will see growing in the next few years as TRT will start to be approved. Oops. So when we have done, you have done all this preclinical study, you have to do your IND, your CMC, of course, and there is questions that are very important to answer with the isotope. The dosimetry is key, which is something that you don't do usually with other kind of therapy. It will be related to the toxicity of your compounds and then the efficacy in humans. Sometimes you have compounds that are very efficient, but if the dosimetry is not favorable, you might not be able to give them enough radioactivity in the tumor if you have too much in the kidney, for example. So it's important to well understand your biodistribution and the ratio between the tumor uptake and other organs to be able to extrapolate in human the dosimetry and further toxicity. So just to summarize, farm image is what we call is a network. At Oncodesign we can perform multi-studies, but we are not a standalone company that we perform everything, and we have developed a very strong network with our collaborator. First in Dijon, that's what we call Farm Image, with the Cancer Center for the phase 1 study. I mentioned Chematech for the chelating agents, some labs that are working with us, and altogether we have a multidisciplinary expertise from chemistry to clinic, and we can clearly help you to move your compounds starting from the chemistry up to the phase 1. We have a multimodality platform with all the technologies requested along the development process of TRT to do all the imagings, and a large choice of models for the preclinical validation and efficacy study. And then when it's starting to go to clinic, we have collaboration with Institut Curie at the university in Germany, in Tübingen, which is a famous center for TRT. And to finish this webinar, some take-home message. So the development of targeted radiotherapy requires a continuum of expertise from the radiochemistry to pharmacoimaging, to integrating drug discovery process and radiotherapy clinical strategies. Combination is clearly of high interest, notably with immunotherapies to enhance immune infiltrate and increase DNA damage in tumor. The selection of preclinical models could be challenging. You have to take into account different criteria, the target expression, of course, Do you want to work with syngenic mouse model versus humanized, depending on your compound, depending on the challenge or the intercross with immunotherapy? Orthotopic versus subcutaneous, it might clearly affect your biodistribution. If you consider the immunofiltrates or the stroma and the mutation loading, that also could modify your efficacy. An imaging companion diagnostic has not to be forgotten. You need to monitor the target expression. You need to have it in parallel, to develop it in parallel of your therapeutic compounds to be able to stratify the patient and assess the therapeutic efficacy. I'm done for today. I hope I give you enough background and for targeted radiotherapy. And if you want to know more, we'll be pleased to discuss at the next ENM meeting, where we are sharing a booth with Kematech, booth 33 at the ENM. And as we like to say, it takes two to tango. You need to, on one side, to have the physics of the isotopes, on the other side, the biologics of your compounds, and this is you need both to be able to make it right. Thank you very much, and if you have any question, I'm pleased to answer them.
Thank you very much, Cyril, for the presentation. Here is the Q&A time, so feel free to use the chat box to ask your questions. Cyril, I already have 2 questions that came up during the presentation.
Yes.
If you're